So I've got 2 pieces of filter paper, which we put in a petri dish and wet. So it just needs to be moist. So remove the excess, then put the cover back on. So I'm just going to take the inflorescences from these Arabidopsis plants, put them onto the moist filter paper so they don't dry out. So these are the components that we need to do the immunolocalisation. So we have our Arabidopsis buds on the moist filter paper. We have the EM digestion mix, which is used to break open the cell walls. The forceps for dissection with a pointed needle, and the brass rod as well to help break down the cell walls. So we have a lipsole for spreading the nuclei, and the glass slides. So the glass slides that I use are frosted Super Plus, as it seems better that the cells actually stick to these glass slides, because with other glass slides often the cells are washed off. So we use a binocular microscope with the LED light source, and also 37 degrees moist box for the digestion mix. So to get the filter paper to the right degree of wetness, I pour off the excess water. So in this state, the filter paper is not too dry, it's not too wet, so it's easy to manipulate the anthers. Okay. Okay. So we start dissecting the inflorescences by removing the buds, the smallest buds first, which are in the centre. So I've made a pile of the smallest buds, as it's the smallest buds that contain prophase 1 of meiosis. So the first thing I'm going to do is to remove the largest buds, which contain pollen. Okay. So I'll see. So now I take a slide, label it on the frosted side. You can tell the frosted side by feeling. So we just label this immuno. So I do ASY1, rabbit, zip1, rat. Just put the date on. 12th. Okay, so to this slide, I'm going to add 5 microliters of the EM digestion mix to the centre. Then I'm going to transfer the dissected anthers to this with a forceps. And using the brass rod, so first I'm going to time this. So I'm going to use the brass rod to gently break up the tissue. Do this for a minute. So then add a second 5 microliters to this. Mix that in. Then add to the 37-degree moist box. I'll incubate for 2 minutes. So the anthers have been in the EM digestion mix for 3 minutes now. So I'm just removing it from there, wiping the underside of the slide. So at this point, add 10 microliters of 1% Lipsol directly to the slide. So Lipsol is very useful to spread the chromosomes and remove the background. So I'm using the rounded needle to mix the Lipsol and the anthers and the chromosomes. Okay, so to fix the nuclei to the slides, we use 4% paraformaldehyde, which is chilled. I'm just going to add this to our mixture directly onto the slide. And then I mix it together gently with the pipette tip and leave that to dry for an hour. So after an hour, the slide is dry and the cells are fixed to the slide. So now what we need to do is add the primary antibodies to the slide. So I've already added ASY1 and ZIP1 to EM block at a dilution of 1 in 500. So now I'm going to add 50 microliters to the slide of this mixture on top of the cells. So to ensure that the slide doesn't dry out, we add parafilm to the slide, like so. And get rid of the air bubbles so the antibodies are in contact with the cells. And we add this to the moist box, put the lid on, and we can incubate this for 30 minutes at 37 degrees or overnight at 4 degrees. So the slide has been incubated for 30 minutes at 37 degrees. And now we're going to wash the slide 3 times, 5 minutes in PBS plus 0.1% Triton in the coupling jar. So now the slide has been washed, I'm going to remove the excess wash buffer and tissue paper. Just dry the underside. And again, I'm going to add the secondary antibodies directly to the slide. And then add the parafilm. so the slide does not dry out. And again, incubate it 20 to 30 minutes at 37 degrees. So after incubating the secondary antibodies, we now wash the slide again in the 1 times PBS and 0.1% Triton. We do this for 3 times 5 minutes. Now the slide has been washed, again I'm going to remove excess wash buffer on the tissue paper. Just try the underside. And then add DAPI to the cells. And a cover slip to the top. And just block this. Now our slide is ready to be looked at.